So, today we're talking about a technology that is just completely changing the game in the fight against cancer. It's called next generation sequencing, or NGS. And, the best way to think about it is like this. Imagine having a tool that can read a tumor's entire genetic code with, breathtaking speed and detail. It's what's letting us turn this one-size-fits-all battle into a truly personalized mission. Okay, so let's just start with the biggest problem, right? Cancer isn't just one thing. It's more like a shape-shifting enemy. It's really thousands of different diseases, and every single one is driven by its own unique brand of genetic chaos. So how on earth do you find the right weapon when every patient's cancer is, down at the molecular level, its own separate thing? For decades, that was the fundamental challenge, and honestly, it was overwhelming. You see, the old methods were like trying to understand a massive, super complex book by just reading a few random words on a single page. They were slow, they were expensive, and they were so incredibly narrow. Yeah, we could maybe spot a well-known mutation here or there, but we were just missing the full story. The context, the grammar, all the plot twists hidden deep inside that tumor's DNA. But then, a revolution happened. A revolution in our ability to read that entire genetic library. And this is where next-generation sequencing, NGS, steps onto the stage and just changes everything. Now, to really appreciate what a huge leap this was, you have to look back for a second. The first generation, Sanger sequencing, which came out in 1977, it laid the foundation. It was brilliant for its time, but man, it was slow. The real game changer was when this second-gen tech arrived around 2005. That's what gave us NGS and this massively parallel approach. And look, this wasn't just a small improvement, it was a completely different scale of operation. And the difference is, well, it's just dramatic. While the original method could only read one single DNA fragment at a time, NGS reads millions of them all at once. This incredible throughput is what makes it so much faster and way more cost-effective for looking at entire genomes. It's literally the difference between a single scribe, painstakingly copying a book by hand, and a high-speed digital scanner digitizing an entire library in minutes. So, how does this magic actually work? I know, it sounds impossibly complex, but at a high level, you can break it down into four main steps. First, you get the DNA out of a sample, like a tumor biopsy. Then that DNA gets chopped up into tiny pieces and tagged. That creates what they call a library. Next, the sequencing machine goes to work, reading all of these millions of pieces at the same time. And finally, really powerful software acts like a master puzzle solver, putting all that data back together to reveal the tumor's complete genetic sequence, mutations, and all. Okay, so we've got this amazingly powerful technology. Now, let's look at how it helps us build a way better diagnostic toolkit. How does reading a tumor's DNA actually help us find those critical clues we need for a diagnosis? Well, it really boils down to three incredible applications. First, getting a complete genetic profile of a tumor that's already there. Second, we can use it to find inherited risks that might make someone more likely to get cancer in the first place. And third, and this one's fascinating, we can use it to hunt down the very last cancer cells that might be hiding out after treatment. One of the single biggest breakthroughs that NGS has given us is the liquid biopsy, and this is a total game changer. Instead of a surgeon having to go in and cut out a piece of a tumor, doctors can now find tiny fragments of its DNA floating in the bloodstream. They call it circulating tumor DNA, or ctDNA, and they can get it from a simple blood draw. This lets them profile a tumor's mutations and track how it changes over time, all without making a single incision. It's incredible. But it goes way beyond just diagnosing a cancer you already have. NGS is a powerful tool for prevention. By screening for inherited mutations in genes you've probably heard of like BRCA1 and BRCA2, it can flag people who have a much, much higher lifetime risk of developing certain cancers. And having that knowledge, well, it empowers them and their doctors to be proactive with closer monitoring and preventative strategies. And just look at these numbers to see how critical this information can be. For a woman with a BRCA1 mutation, the lifetime risk of breast cancer jumps from about 13% for most people all the way up to 72%. And for ovarian cancer, it's a staggering 44%. Knowing that information completely changes how you approach your own healthcare. So what about after treatment is over? A patient might look like they're in remission, but there could be a few cancer cells just lingering. This is called minimal residual disease, or MRD. The incredible sensitivity of NGS lets us spot these last few hidden cells in a way that older methods just couldn't. It gives us a crucial early warning if the cancer is even thinking about making a comeback. All right, so all of this super detailed diagnostic information leads us right to the ultimate goal of modern oncology: personalized treatment. It's all about moving away from treating lung cancer as one disease and moving toward treating your specific lung cancer based on its own unique genetic instruction manual. And hey, this isn't science fiction, okay? This is happening right now. About 10% of all FDA-approved drug labels already have information that links how well they work to a patient's own genetic data. This technology is being woven into the fabric of standard medical care as we speak. So how does it actually work in the clinic? Well, NGS helps us find the exact driver mutation, that one specific genetic mistake that's basically telling the cancer cells to grow and divide out of control. So, for instance, if we find a mutation in a gene called EGFR in a lung tumor, we can use a drug that was specifically designed to shut down that one faulty signal. We're not just carpet bombing the tumor, we're switching off its engine. And this really gets to the heart of the difference. Think of traditional chemotherapy as a blunt instrument. It attacks all fast-growing cells, whether they're cancerous or healthy, and that's why it causes such tough side effects. Targeted therapy, on the other hand, which is guided by NGS, is more like a sniper rifle. It zeroes in on the very specific molecular pathways that are broken only in the cancer cells, leaving most of the healthy cells alone. NGS has already completely changed cancer care, but believe me, this story is far from over. So let's take a quick look at what's coming up on the horizon for genomic medicine. I mean, the future here is incredibly exciting. We're moving towards something called single-cell sequencing, which will let us see the genetic differences within a single tumor. We're also working on integrating all this genomic data with other massive data sets to get an even clearer picture. But of course, big challenges are still there. Making sense of this absolute mountain of data takes huge amounts of computing power and expertise. And of course, we have to navigate the huge ethical questions and the vital need to protect this deeply personal patient information. So, let's end on this thought. We are getting closer and closer to a future where sequencing a tumor's genome is as routine as getting a blood test. The question then becomes, what does medicine even look like when we hold the complete biological blueprint for every single patient's disease? The possibilities? Well, they're as vast as the genome itself.